All right, so we broke down our glucose in the past few sections here, and we extracted some energy, but most of that energy actually got stored in high energy electron carriers. In chapter 7.4, we are going to talk about oxidative phosphorylation, which is going to allow us to extract the energy out of those. And to do that, we need to talk about this system called the Electron Transport Chain. Sometimes it's called the Electron Transport System. And this is going to be used to produce a proton gradient. Remember, protons are just hydrogen ions, and they will be used to generate a large amount of ATP. So this enzyme that we'll talk about will be very important. Okay, let's get into it. So to review, we had glycolysis that took glucose and broke it in half. We had our 2-pyruvic acid. We produced 2 ATP. We sent the pyruvic acid to the mitochondria, where they got converted into acetyl-CoA. We cut one of the carbons off, producing CO2. Then we sent it through the citric acid cycle and cut the other 2 carbons off and produced CO2 from that as well. We did make 2 net ATP, and the main thing, though, that was produced here is NADH and FADH2, our high-energy electron carriers. These high-energy electron carriers will be utilized in the next step, the electron transport chain. So what is the electron transport chain? It is a system that uses the energy from those high-energy electrons to pump hydrogens out of the mitochondrial matrix into the intermembrane space. Remember, there's 2 membranes in there. The space between the 2 membranes is called the intermembrane space. So we're going to create a hydrogen ion gradient. Now, what powers this? What pulls these electrons through this system is oxygen in aerobic respiration. So oxygen is called the terminal electron acceptor in aerobic respiration. That means that oxygen, which is down here, is pulling the electrons through this system, and in the process, hydrogen ions get pumped across the membrane. So we said, why do we need oxygen to live? Well, this is the answer. We need oxygen to pull those electrons through the electron transport chain. So let's go and watch an animation of this.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits two high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in two important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
All right, so obviously that animation has a lot more information than we fully need here. Let's break down the basic parts of what's going on here. Okay, so we have our mitochondrial inner membrane. We have the matrix here and the intermembrane space over here. In previous steps of breaking down glucose, we harvested energy in our high energy electron carriers, NADH and FADH2. When those high energy electron carriers reach the electron transport chain, the enzymes in there are all pulling the electrons from these things through the chain to oxygen. Oxygen is really the thing at the end that's grabbing hold and pulling those electrons towards it. It wants those electrons. In the process, hydrogen ions, protons, are pumped into the intermembrane space. We call this process chemiosmosis because it's kind of like osmosis, but instead of water, we're moving a different chemical. This hydrogen ion gradient forms a high concentration in the intermembrane space. We're then going to allow it the chance to flow back through to the lower concentration. We're going to do that, though, by sending it through an enzyme called ATP synthase. ATP synthase does what its name says. It synthesizes ATP. So it takes ADP and inorganic phosphate and joins them together using the energy from this hydrogen ion gradient, forming ATP. This is the step in the whole thing that generates lots of ATP. This hydrogen ion gradient, we call it a proton motive force. In bacteria, they do the same thing. They don't have mitochondria. They do it with their membranes. But this proton motive force can do many other things. It can even move the cell in some cases. But in eukaryotes, in the mitochondria, this is generating lots of ATP. ATP synthase is a super cool molecule. We kind of saw it a little bit in the animation, but it has an intermembrane bit, and it has a stator, which is static, and then it has this rotor here that turns. And the flow of hydrogen ions through that allows it to catalyze the production of ATP. It is made up of many, many different subunits. You can see them all here: alpha, beta, delta, beta 2, the C unit here, all these different subunits of it, the gamma unit there, the epsilon unit there. It is a well-studied enzyme, but it is super critical because it makes the majority of our ATP. I just think it looks super cool. It's like this little motor. It's almost like a hydroelectric generator, right? It's very much a cool looking enzyme. So let's look at what happened in this step. Well, so we used our high energy electrons to pump hydrogens into the intermembrane space. That hydrogen ion gradient, that proton motive force as it's called, flowed through ATP synthase, and in the process generated 32 ATP. Compared with 2 and 2 here, that's a lot. That's what I want you to know, right? A lot got generated there. We did utilize oxygen in the process. We had to put oxygen in to draw the electrons through the electron transport chain. So, overall, how much ATP can we get from one glucose molecule? This is a theoretical equation. It's not quite this perfect. There is loss in a real biological system, but so we can take one molecule of glucose and our oxygen. We will cut the carbons off of glucose and produce CO2 in the process. Water gets made as a byproduct from this reaction, but we use that energy to generate 2, that's 4, and 32, that's a total of 36 potential ATP. How much is 36 ATP? Well, it's really actually not that much. So if you want to take one amino acid and another amino acid and stick them together, that generally requires the energy from one ATP. Now consider your average protein is several hundred amino acids long. You have to get a lot of energy just to survive, right? Just to do the chemical reactions that are occurring in you, let alone move and think and do all these energy intensive activities. So you need a lot of glucose, right? Glucose is not the only place you can get energy, though. You're probably aware that you can eat other sugars like fructose, right? Fructose can feed into this pathway. Here's glucose being broken down. Fructose can feed into a side pathway and can get broken down as well. So we can utilize other sugars in the process. So it is not just glucose that gets used, but we have focused almost entirely on glucose because it's the simplest example here. If you went into nutritional biochemistry, you would learn all about the other pathways for breaking things down. We'll talk slightly about like fats and proteins, how they can feed energy in a later section here. All right, so we're done extracting all that energy out, right? There were many steps to it. Let's go ahead and review the steps here. So this was the process of aerobic respiration, which involves oxygen. So I would like you to put the correct order of steps together for the breakdown of glucose for a human doing aerobic respiration. Go ahead and pause the video. Think about this one. Hopefully, you either remember the first or the last reaction. That could help us immensely. I always remember the first one. Glycolysis is where we start. That's breaking sugar in half. So we already nail it down to two options, right? A or B. So glycolysis is our first. What's the last one? That might be an easy place to think about. Well, the last one is oxidative phosphorylation, where that oxygen was finally used. So that's of no help to us here. So unfortunately, it's going to be memorizing the middle bit. So what happens after glycolysis? Well, we take our pyruvate and we turn it into acetyl-CoA, cutting one of the carbons off. So acetyl-CoA generation is next, and then it goes into the citric acid cycle. That all checks out. So A, glycolysis to acetyl-CoA generation to the citric acid cycle to oxidative phosphorylation is our correct order for this. All right, so oxidative phosphorylation. That's where we'll fully extract the energy that's been transferred into the high energy electron carriers, NADH and FADH2. The electron transport chain pulls the electrons through, and oxygen is the terminal electron acceptor. It pulls on the electrons the strongest. That's in aerobic respiration, though. I'll talk about when oxygen isn't present in a moment. The membrane protein pumps in the electron transport chain pump hydrogen ions, protons, from the mitochondrial matrix into the intermembrane space. That proton gradient builds up, and then it's allowed to flow through ATP synthase, turning the enzyme and adding a phosphate to ADP to form ATP. This process is called oxidative phosphorylation because we're phosphorylating that molecule. The theoretical max ATP from one molecule of glucose is 36. So there were 2 from glycolysis, 2 from the citric acid cycle, and a lot, 32, from oxidative phosphorylation. So you can see if you don't do this bit, you don't get very much energy from glucose. Okay, in the next sections, we'll talk about some alternative ways of extracting energy here.